Good day, my name is Maya Erna, and today I would like to present to you a research paper entitled De Novo Whole Transcriptome Profiling of Edwardsiella tarda isolated from infected fish, Labeo catla. For introductions, as we know that transcriptome is a study related to RNAs such as expression, function, location, trafficking, degradation, as well as transcriptional modification. The paper focused on the Edwardsiella tarda bacteria, which is a gram-negative bacteria that cause infection in human and fish. It is first time isolated in year 1962 from the Japanese eel in Japan. It is a major pathogen for the freshwater and marine fish that cause Edwardsiella disease that lead to the mass mortality in fish. The bacteria usually infect the fish by entering through skin, intestine, or gill. The research is important to find some novel genes and their biological function to understand the mechanism behind the bacterial infection at transcript level. The objective is to analyze the tarda transcriptome sequence data isolated from the fish and then to discover the significant genes and their interaction pathway in understanding the relationship between the transcriptome and the phenotype of the tarda. The figure shows the methodology flowchart for the research. After bacterial culture, the RNA was isolated using the RNA isolation kits, and the library preparation were done using the solid total RNA sequence kit. The assessment of the total RNA integrity was done using the bioanalyzer. The sequence then were assembled by the de novo assembly using the Velvet software and Oasis Part 9 before the transcript contigs were mapped. The transcript contig obtained was used for functional annotation by aligning them with the bacterial non-redundant dataset from the NCBI using the BLASTX. The genes was then identified using several software and databases such as SwissProt, Unitremble, BLAST2Go, and QuickGo. The significant pathways of tarda infection can be obtained using the KEGG database. And the protein-protein interaction can be predicted using string database. Results and discussions. After assembled transcript, a total of 9,657 annotated transcript contigs were mapped on the GO database, and from there only 6,749 were GO annotated. From the GO results, it showed that the expressed genes were mostly functionally used for the molecular functions, cellular component, and biological process. The figure showed the functionality distributions of tarda transcript by gene ontology analysis. We can see that biological process distribution involve gene related to such as immune system and metabolomic process. For cellular component distributions, involve gene related to such as organelle, membrane, and cell part. And for molecular function distributions, involve the gene related to binding and catalytic activity. For the KEGG pathway analysis, the study found that most significant pathway involved in biosynthetic pathway, immunology diseases, metabolism, degradation, infection, and transport. Here is the figure of KEGG pathway distribution analysis. We can see that several significant pathway group distributions, such as for immune disease pathway in red and biosynthesis pathway in blue. For the protein network analysis, it is mentioned that the protein involved in the tarda infections are virulence-related membrane protein and flagellin domain containing protein. Other protein-protein interaction that mostly expressed are such as protein involved in metabolic pathway, flagellar assembly, microbial metabolism, and carbon metabolism. As for the conclusion, this paper has successfully carried out a de novo whole transcriptome sequencing of tarda isolated from infected fish using the solid sequencing platform. The transcriptome data analysis performed using this bioinformatic outline had identified a few immune-related genes, examined the GO analysis, studied the functional gene enrichment, and as well as protein network analysis to understand the expressed protein correlation. The result in this research can provide a deep insight into the transcript level analysis of tarda infection that will be helpful in identifying reliable therapeutic target in the near future. Thank you.